So in this chapter, see this overview of body's amino acid pool, sources and utilization. This table is important. Okay. So that means the body, like body protein, how much it is? 10 to 12 kg. Our suppose if my body weight is 50 kg, so among that 10 to 12 kg of body weight is contributed by protein. Basically, proteins are not stored in the body. Remember that. They only
Yes.
form structural components, like hair is made up of protein, keratin, isn't it? Nail is made up of protein, keratin, isn't it? So like muscles are protein. So like bone is also a protein. So we have proteins are generally making a structural framework of the body, but they are not stored in the body. So suppose if we take more protein also in the diet, it will be used in the body building mechanism, and rest of the protein will be excreted in the urine in the form of urea, isn't it? So like protein breakdown, how much? See, 350 to 400 grams per day. So that is like body amino acid pool, it will be utilized like it is somewhere it comes to 100 grams. So if it is more, though you have taken like some protein in the diet and around 350 to 400 grams and amino acid pool, it has been diverted, and after that whatever has not been utilized by the body, that is lost, protein loss, that is 30 to 50 grams mostly as urea in the urine. Okay, so this is the like cycle, like the protein cycle what we have now. Not only that, see, our body is also utilizing the protein what we take in the diet for the purpose of protein synthesis, nothing but for the making of muscular system. Okay, for the formation of hair, nail, whatever protein material is there in our body, that is being diverted towards synthesis, that is protein synthesis. How much? Around 300 to 400 grams. Okay, then you see like synthesis of non-protein compounds is also being taken place by this body amino acid pool. Like 30 grams per day, it is utilized for the formation of creatine. Creatine is muscle protein which is utilized for the formation of ATP. We will study that creatine is converted to creatinine. Okay, and there you will see ATP will be generated. Porphyrins we will see in the formation of hemoglobin. Phospholipids, okay, purines
Lipid.
we see in the nucleoproteins. Purines and pyrimidines, they are also one form of protein. So body protein is being utilized for the synthesis of all these byproducts of proteins. Okay, proteins can be also converted to carbohydrate and fat. If you remember, there is a cycle called as gluconeogenesis, isn't it?
Yes, ma'am.
That is the formation of glucose from non-carbohydrate source. That means carbohydrates actually are formed from proteins. Okay, and there are certain enzymes what we usually study in gluconeogenesis. So similarly, proteins can be also converted to fat. Now, proteins can be also used for the production of energy, but very rarely. Suppose if we don't take carbohydrate in the diet, suppose if we don't take fat in the diet, then body protein will be mobilized to give energy. Like you see in the severe cases of kwashiorkor and marasmus.
Yes,
Marasmus,
ma'am.
how skinny child you will be seeing, isn't it? So what does it
Yes, ma'am.
mean? There is nothing to eat. They are under severe starvation. So whenever a body is under severe starvation, it will start utilizing its own body proteins. Okay. So energy like 10 to 15% of body's daily requirement, it can be like coming from proteins. Okay, that is utilization. So I hope you understood. No, but this part.
Yes, ma'am.
So dietary protein, we take 40 to 100 grams. Synthesis of non-essential amino acids also can take place from this body amino acid pool, okay, from various sources. So this is overview of body's amino acid pool. Body's amino acid pool is coming from the protein what we take in the diet, and the weight of body's amino acid pool, it can be converted to like non-protein compounds. It can be converted for carbohydrate fats. It can be utilized for the production of energy.
Energy.
These are all. Excess protein can be just excreted in the urine in the form of urea. Okay, so this is like an overview, like what is happening to the protein, what we take in the diet. Okay, beta? See,
Yes, ma'am.
no storage form of amino acids, as I told you. There is no storage at all. So utilization of amino acids from body pool. Now, metabolism of amino acid. See here, even this is not so important, but transamination, deamination is of course very important. We will study in detail. Now, what is transamination? Here, we have around 20 amino acids, no, Shubhangi? Standard, usually when we talk.
Yes.
Of course, many new amino acids are being discovered, and some of the biochemists say we have 22, 25, something like that. But usually, standardly, we say like 20 amino acids are there. So these amino acids, suppose if they are transferring the amino group to one keto acid, then you call it as transamination. Transfer of amino acid, like amino group, from an amino acid to keto acid is known as transamination. Now see here, this is one amino acid. Think it can be a glycine, lysine or anything. And this is some keto acid. So keto acid, like here you have functional group CO. Here amino acid functional group is NH3. Yeah, ionic form, ammonium. So you are finding this amino acid from this glycine, for example, is being transferred to keto acid. Now keto acid becomes amino acid becomes keto acid. Isn't it, Shubhangi?
Yes, ma'am.
The amino group has been transferred. That's the meaning transfer of amino group from the amino acid, like keto acid, but transamination. We require enzyme transaminases, and the like PLP is like it's a like pyridoxine. No, so it is acting as coenzyme. Okay, pyridoxal phosphate. It's a coenzyme which is actually enhancing the speed of the reaction. Okay, so that is transaminase is converting the amino acid transfer from amino acid to keto acid. That is the summary. Okay, so the same thing has been elaborated here. So just draw this picture, and one more like example has been given where like the examples of amino acids have been given. Now the amino acid which has been given here is alanine. So the amino group from alanine is now being transferred to pyruvate. Pyruvate is the keto acid. Alanine is the amino acid. So amino group is being transferred from alanine to pyruvate. Hence now you see there is like what is the enzyme? We usually say transaminase. Now to be very specific, it is glutamate, pyruvate, transaminase. So this amino group is now being handled by PLP, pyridoxal phosphate, I said just now, coenzyme.
Yes, ma'am.
Once this pyridoxal phosphate receives this amino group, it forms pyridoxamine phosphate. Shubhangi.
Yes, ma'am.
So this pyridoxamine phosphate is now converting, see this amino group is now being gradually transferred from this pyridoxamine phosphate. From alpha ketoglutarate, it is now being transferred to glutamate. Okay, you can see the amino group is transferred. That means from alanine, it is transferred to pyruvate. One more example has been given from alpha ketoglutarate, it has been transferred to glutamate. Okay, so that
Yes,
is
ma'am.
transamination. Just give this example, beta, and same thing has been put in words. Read once, you will definitely understand. Okay, and
Yes,
this
ma'am.
enzyme PLP shift base, you just exclude it. You need not, because this is practicals when you do know. It comes under practicals, not theoretical part of the subject. Okay, beta?
Yes, ma'am.
Skip this. Okay, then. Now coming to deamination. So deamination in the sense, see this is the amino, like structure of one amino acid, right? Like glutamate, it has amino group. So if through series of reactions, this amino group will be removed from amino acid. The removal of amino acid is nothing but deamination. So what is the purpose? Why this amino group has to be removed? The amino group has to be removed because it has to be channelized to enter into urea cycle. Once the amino group like ammonium ion is freed, then only it will combine with carbon dioxide, finally to form the end product urea. That is the purpose of these two steps. Basically, sometimes question will be asked like discuss transamination, deamination, and then how this ammonia is channelized. Okay, for the purpose
Yes,
of
ma'am.
detoxification. That's a hint like you have to write urea cycle. Okay, beta? So now see here,
Okay.
like we will go with deamination now. So again, you have to learn this everything. Like of course here, if you can remember the formulas, it's well and good because you are actually highlighting the removal of this group amino, isn't it? NH3 ammonium ion. Okay, so L-glutamate, this ammonium ion in the presence of NAD, NADPH, is converted to alpha aminoglutarate. From here, it is converted to alpha ketoglutarate. Both the reactions are oxidation reduction reactions, which are catalyzed by glutamate dehydrogenase, GDH. Finally, L-glutamate is converted to alpha ketoglutarate plus ammonium ion. Okay, beta? So that is
Yes, ma'am.
deamination. So in this cycle, you are finding the amino group has been removed. Removal of amino group through this series of
No.
steps. That is deamination. Because here NAD, NADP, biological oxidation, that means like transfer of hydrogen ions or removal of oxygen or addition of oxygen. We generally use these two definitions for oxidation reduction. So whenever NAD, NADP is involved, always remember it comes under oxidation reduction reactions and you call it as oxidative deamination. Okay,
Yes, ma'am.
oxidative deamination. So we also have like some non-oxidative deaminations also. Few examples you can give. Directly few amino acids are given like serine, threonine, homocysteine. Here enzyme dehydratase is acting and the ammonium molecule is liberated. So this amino acid is now converted to respective keto acid. Okay, beta?
Yes, ma'am.
Similarly, many examples are given like cysteine is converted to pyruvate, histidine is converted to urocanate. All hydrolyze, like that means removal of water molecule, like is like dehydratase here. Okay. Then here you have again, see all hydrases in the sense that water molecule will be removed.
Removal of water.
And you will see amino group will be now converted to keto group. Finally, this ammonium molecule is liberated. What we want is NH3 only. Once this ammonia NH3, it's very toxic. If it is remaining in the blood, person will go into coma. Isn't it very dangerous? That's the
Yes,
reason
ma'am.
kidneys are failing, and if kidneys are unable to handle, if liver is failing and liver is unable, formation of urea takes place in the liver. Excretion of
Yes.
urea takes place by kidney. Both are very important. Suppose if liver is failing, what happens? Urea's levels keep on increasing in the blood and person will go into coma and he will die only. Such dangerous end product is ammonia. Okay, beta? So metabolism of
Yes, ma'am.
ammonia. So ammonia exists as this ionic form NH4 plus, that is ammonium ion. Now this formation of ammonia we have seen through two mechanisms, no? Deamination. First
Yes.
transamination, then deamination. Under deamination we have seen oxidative deamination and non-oxidative deamination. These are right.
Yes, ma'am.
Of course, we know like ammonia is very toxic, harmful to brain, and if it is not controlled, person will die. All those things. Now you have to go to urea cycle. So in this urea cycle, you have to learn all these steps better.
Yes, ma'am.
Basic thing is carbon dioxide is combining with ammonium ion in the presence of N-acetyl glutamate. Okay, so 2 ATP molecules carbamyl. Just go through this cycle, but this I'll just attend call. Okay.
Yes, ma'am.
So, and you have certain defects also, no? That also you should learn better.
Yes, ma'am.
This cycle is very, very important. Metabolic defects in urea cycle. So what is the defect? That is the name of the disease we have, and what is the deficient enzyme? Like
Enzyme.
hyperammonemia type 1. The deficient enzyme is carbamyl phosphate synthase 1. So you have to go to the cycle and check like what it is catalyzing. So that means basically it is converting this carbon dioxide plus ammonium to carbamyl phosphate. Suppose if this enzyme is missing, what happens? Carbamyl phosphate is not formed. So in the blood,
Not from.
carbon dioxide and ammonium levels will increase, no? So that will lead to like clinical symptoms like whatever have been given here, just go through once better because nowadays we are getting clinical type of questions only.
Yes, yes, ma'am.
So sometimes we may get question like what is non-protein nitrogen? Okay. So what does that mean? Urea, creatine, uric acid, peptides, amino acid, etc. All these, they come under non-protein nitrogen. Okay. So,
Yes, ma'am.
concentration in blood is 20 to 40 milligrams. The molecular weight of urea is 60 and half of it is 28. So, blood urea nitrogen. See, molecular weight of urea is 60 and half of it, that means is contributed by 2 nitrogen atoms. That means when you see the structure of urea, we have 2 nitrogen atoms. Okay, thus in blood urea 60 milligrams, then half of it around 28 milligrams. So just blood urea nitrogen may be asked better. So blood urea nitrogen is nothing but half of non-protein nitrogen. Okay. So if non-protein nitrogen is 60, half of it, that means it comes to around 30. I think but
30.
28 milligrams is the blood urea nitrogen. Okay. So now coming to individual amino acids metabolism, that is very, very important. Like glycine. Glycine can be an essay. So from glycine, what are the various metabolic things which are produced? You should know this flowchart. From glycine, proteins are synthesized. Glycine can form serine, oxalate, glucose, ammonia. Okay, it can go into one carbon pool. Okay, glycine can be converted to purines, glutathione. It also goes in conjugation for the formation of bile acid, for the production of heme, creatine, everything. So this is
Yes, ma'am.
overall things which can be synthesized from glycine. Okay.
Yes.
So degradation of glycine, all this you don't read, but there is one big cycle. From there you can write everything. This one. Here we have synthesize and breakdown together. You call it as metabolism of glycine. If you learn this, you will be able to write everything.
Yes, ma'am.
See, suppose here you can see two-way reactions are shown. Most of the reactions are two sides you can see.
Two sides.
So glycine, suppose if you start with this first one, carbon dioxide plus ammonium ions together when they combine, okay, it helps in the formation of glycine. We require the enzyme glycine synthase. In the oxidation reduction reaction is taking place. Okay, in the presence of tetrahydrofolate, glycine can be again broken down to carbon dioxide plus ammonium. One carbon metabolism. So whenever one carbon is required during various metabolic processes, glycine can donate one carbon atom from it. Okay, from
Yes,
this
ma'am.
COO. You call it as one carb. Wherever N5N10, methylene tetrahydrofolate, such words come, you remember they help in one carbon metabolism. Once you finish this one, then go with this button. See, glycine can be converted to glyoxylate in the presence of glycine oxidase. Glyoxylate can be converted to glycine by transamination in the presence of pyridoxal phosphate. Okay. Once
Yes.
glyoxylate is formed, it can be further broken down to oxalate and formate. Whenever this word formate comes, you have to remember it is one carbon. You can see in the structure also. Structures you need not remember. Formate, it is undergoing like in the presence of tetrahydrofolate, it forms N10 formyl tetrahydrofolate. This goes into one carbon pool, one carbon metabolism. Now, similarly, again, you have two-way reaction here. Glycine is now converted to serine. So, in the presence of serine hydroxymethyl transferase, serine can be converted to glycine. So, like that. Now, glycine can be also converted. No, threonine can form glycine. Okay.
Okay, ma'am.
So, serine, then we have from serine, you are seeing the formation of ethanolamine. Whatever is given in this flow chart, beta, just try to convert it in the form of words and present in the answer. Okay. So that is all about glycine metabolism. Metabolism in the sense here both synthesis is involved and breakdown is involved. Okay. Anabolism is synthesis. Catabolism is breakdown. Anabolism plus catabolism together you call it as metabolism. That means you are finding both the things here. Synthesis is also shown, breakdown is also shown. Hence you are using the word metabolism of glycine. Okay. See, this is also important beta. Creatine, how much is there in the blood? Okay, normally in the serum and also in the urea. It will be asked for 3 marks question. Okay, creatine and
Yes.
creatinine. Clinical importance. Whenever these levels are increased, it shows like there is something wrong with the kidney. Okay, so that's what we do renal function test to check creatinine clearance test,
Create.
isn't it? So
Yes,
whenever
ma'am.
kidneys are failing, you will see creatinine will not be excreted, it will be retained in the blood. That we will study separately. Glycinuria. That means what? Glycine concentration is normal, but very high amount of it is excreted in urine. Glycinuria. That means in the urine, glycine output is increased. Okay? Because glycine is not being reabsorbed by the renal tubules. It's being excreted by the nephrons. Defective renal absorption is resulting in glycinuria. So this will lead, it will have a tendency for the formation of renal stone made up of oxalate. Okay. Then primary hyperoxaluria, increased urinary oxalate. The urine oxalate levels are increasing, which is leading to oxalate stones. Okay. Then
Yes, ma'am.
basically B6 deficiency, you will see the oxalate levels are elevated. So whenever oxalate levels are elevated, it will lead to the formation of stones, crystallization. Leads to formation of oxalate crystals. Okay, so that can be actually prevented by giving B6 in the diet as a supplement, at least oral medication. So that is B6 administration will reduce the formation of oxalate stone. Okay. But if it is
Yes, ma'am.
endogenous, that means something goes wrong. As such, also our body's metabolism has got deranged, and if oxalate levels are increasing, then even if you give B6, it will not work. Okay, unless if it is from outside, oxalate intake is more, and if you are taking B6, then stones will not be formed. But something has gone wrong and oxalate levels are increasing in blood, then it will lead to formation of oxalate stones. Okay.
Yes, ma'am.
Then phenylalanine tyrosine, very, very important. Again, you should first know phenylalanine, it's an essential amino acid. From here, tyrosine is synthesized, which is non-essential amino acid. Essential amino acid is the amino acid which we need to take in diet. We have a code to remember, AV Hill MP TT. P is actually standing for phenylalanine. Okay. So
Yes, ma'am.
automatically, if you take phenylalanine will synthesize tyrosine. Okay.
Yes,
So from phenylalanine, proteins are synthesized. From phenylalanine, tyrosine is synthesized. So tyrosine can form proteins, glucose, fat, and it can also synthesize pigments like melanin, which are responsible for pigmentation, dopamine, which is a neurotransmitter in basal ganglia, isn't it? Norepinephrine, epinephrine. These are the hormones coming from adrenal medulla, and thyroid hormones T3 and T4. That's the reason phenylalanine and tyrosine are very, very important. Okay. First thing is how the phenylalanine is converted to tyrosine. So here you will see phenylalanine is converted to tyrosine in the presence of phenylalanine hydroxylase. Okay. So here,
yes,
tetrahydrobiopterin is converted to dihydrobiopterin, and you are finding 2 hydrogen molecules are actually taken from this tetrahydrobiopterin. So this is oxidation reduction reactions. Okay. So phenylalanine is converted to tyrosine. Here red mark is shown. Suppose if phenylalanine hydroxylase enzyme is not available, phenylalanine cannot be converted to tyrosine. So this condition is called as phenylketonuria. Phenylketonuria. Okay. Phenylalanine levels will increase in the blood. Tyrosine metabolism, if you can read, you read, otherwise you can leave also. But 99.9% cycle is not important. But if you have time, then you can.
okay, ma'am.
Huh? But this is very, very important.
Yes, ma'am.
Albumin, formation of melanin pigment. Tyrosine is converted to 3,4-dihydroxyphenylalanine in the presence of tyrosinase. Again, that is nothing but dopa. Dopa is converted to dopaquinone, again in tyrosina's presence. Then dopaquinone is now converted to leucodopachrome, okay, and cysteine. So through series of steps, you are finding like the formation of melanin pigments, which are basically black in color. So this melanin pigment is actually responsible for the pigmentation of our skin. Suppose if this melanin pigment is not synthesized, then it will lead to a condition called as albinism.
Albinism.
Very good. Okay, albinism, hypopigmentation.
Yes, ma'am.
That is one thing we should know, and thyroid hormone synthesis. Again, this flowchart you have to memorize. Thyroglobulin. So this is the tyrosine, which is an amino acid, which is now like utilizing iodine molecule. Okay, and it is forming, if one iodine molecule is utilized, it forms monoiodotyrosine. If 2 iodine molecules are utilized, it forms diiodotyrosine. Okay, MID and DIT. So we have T3 and T4, right? For T3, diiodotyrosine plus monoiodotyrosine T3 forms. If diiodo plus diiodo, that means 2 iodines plus 2 iodines, then it will lead to the formation of thyroxine. Very good. Okay. So this flowchart you have to remember, beta. Then comes
Yes,
this one, catecholamines. So tyrosine is converted to again dopa to dopamine. dopamine to norepinephrine, norepinephrine to epinephrine. Here, actually, this last step also is useful when we do one carbon metabolism or sulfur containing amino acids, SAM, active form of
Sam,
methionine.
methionine. Yes, ma'am.
Very good. S-adenosyl methionine. So this S-adenosyl methionine, this is methyl group is now donating the required carbon atom. One carbon transfer is taking place. Here you are finding we have only 2 carbons for norepinephrine, whereas for epinephrine we have 3 carbon atoms, right? So that carbon is donated by SAM, S-adenosyl methionine, which is active form of methionine. Okay, this example you can write when we do sulfur containing amino acids like methionine. Okay, so this whole
Yes.
part is important. Beta. Functions of catecholamines, we know these are nothing but medullary hormones which act as neurotransmitters which help us to cope up with stress, isn't it? They prepare
Yes, ma'am.
us to face any situation, isn't it? Those are catecholamines. Dopamine, and dopamine is a neurotransmitter which is basically synthesized in basal ganglia of the brain. Suppose if dopamine is not synthesized, then it will lead to a condition.
Parkinsonism.
Very good, Parkinsonism. So Parkinson's disease, here what we will give, levodopa is the drug of choice, right?
Yes,
L-dopa,
ma'am.
L-dopa, levodopa. Why? Because we know like directly if you give dopa, dopa will not cross the blood-brain barrier, whereas levodopa will be able to cross
Cross.
blood-brain barrier. And once it enters the basal ganglia area, levodopa will be converted to dopamine, where it will be utilized as a neurotransmitter, rectifying the defect. Okay, so that is
Yes, ma'am.
disorders of tyrosine, phenylketonuria. So phenylalanine hydroxylase enzyme is missing, hence phenylalanine cannot be converted to tyrosine. Okay. So what happens? Suppose if such a thing, if something goes wrong like that, so baby will have mental retardation. Baby will not be able to walk, talk, failure to grow, fits, tremors, almost mental retardation. Phenylalanine starts accumulating in the brain. That's the reason all these defects will happen. Then synthesis of serotonin from tryptophan is insufficient. This is due to competition of phenylalanine and its metabolites with tryptophan that impairs the synthesis of serotonin. That means serotonin
Yes,
and
ma'am.
phenylalanine both are utilizing the same enzymes. So all enzymes are now being utilized towards phenylalanine. Hence, serotonin will not get a chance for the formation. Then defect in myelin. Myelin sheath is not formed. Myelin sheath, we know now it helps in quick conduction of impulses.
Yes, ma'am.
In case of phenylketonuria, you will see even myelin sheath production becomes defective. And that is one defect what you study under phenylketonuria. Okay, once you are done with that, there is one more defect, that is effect of pigmentation. So we know melanin is synthesized from this tyrosine only. So suppose if this is melanin pigment is not synthesized, we know it leads to hypopigmentation. So skin becomes white, isn't it? Even
Yes,
hair
ma'am.
also become white. Even in the iris, pigments are not there, isn't it? So diagnosis of phenylketonuria, generally we check the urine and the high levels of phenylketonuria. Phenylalanine levels will be high in the urine and it will be excreted in the urine. So Guthrie's test is like we will just check with this Bacillus subtilis. If phenylalanine is available, it will be using it as culture media and this Bacillus will grow as food, Shubhangi.
Yes, ma'am.
If this bacillus is growing, that means person is suffering with phenylketonuria. Okay. If this bacillus colonies are not formed and this bacillus is not growing, that means the urine has no phenylalanine. Okay.
Ellen.
So that is called as Guthrie's test. Okay. There is one more
Yes, ma'am.
test that is like we can also check the presence of phenylalanine with the help of this ferric chloride. Okay. So where suppose if you just treat the urine which has phenylalanine with this ferric chloride, you will see green color is obtained. Okay.
Hmm.
Of course, but many false positive tests are there. Hence, this Guthrie's test is more standard when you compare with this ferric chloride. So in this case, like phenylketonuria, so what should we do? See, plasma phenylalanine concentration within normal range. So we have to see like diet is not containing phenylalanine, Shubhangi. Okay, we have to
Yes, ma'am.
milk formulas designed in such a way, milk powders will be there where you will see phenylalanine is avoided. Hence, it will not be converted to tyrosine, and all these phenylketonuria and all such defects we can prevent. Okay, complications because very dangerous. No, it is also causing
Mm.
mental retardation, which is seizures and all. So, in order to prevent that, we have to give mother's milk has to be like
Given.
avoided. Don't give mother's milk because it contains no phenylalanine.
Oh,
Instead,
yes, ma'am.
milk formulas we have to give.
Hmm.
Special milk formulas, milk powders will be available. Lactogen 1,
Yes, ma'am.
2, 3, some dabbas will be there. So special milk formulas are designed to treat phenylketonuria. Okay. So for few months till the baby is stabilized. Okay, till like myelin sheath is formed, like everything. Baby is able to walk, talk, and all. We have to protect. Okay, prevent mother's milk and give like milk powders instead. Okay.
Hmm. Yes, ma'am.
Neonatal, that means from in the newborn baby itself, you are finding tyrosine levels are increasing in blood. You call it as neonatal tyrosinemia. Okay, that means tyrosine levels, tyrosine production is increased. Okay, because of the defect of this enzyme P hydroxy phenyl pyruvate dioxygenase. Okay, then
Yes, ma'am.
alkaptonuria is also very important beta. So black or Coca-Cola colored urine, you will see alkapton. Brown or black colored urine, Coke colored urine. So, which enzyme is defective? Homogentisate oxidase.
Oxidase. Yes, ma'am.
There is a flow chart for this. This one. See, you are finding, see, alkaptonuria in the sense homogentisate should be converted to 4-mallyl acetoacetate. So in the presence of homogentisate oxidase, suppose if this enzyme is missing, you will see homogentisate concentration will increase in the blood and same thing will be excreted in the urine. So that condition is nothing but alkaptonuria. And if the
Urea.
urine is standing for a longer time, you just allow it to stand in a test tube for a longer time, it will get converted into black color or coke color. Okay, brown color. Because you will see it leads to the formation of quinones. Okay, corresponding quinones are formed, which will give that particular discoloration of urine. So,
Yes.
so suppose if this chemical, the end product, alkapton, if it is there in the blood, it starts getting deposited in the connective tissue, bones, various internal organs, outside also, nose, ear. So there also you will see typical pigmentation will be seen. You call it as ochronosis. Okay.
Okay.
Deposition of this homogentisate, it will lead to joint pains. Okay, nothing but arthritis. Okay. It's very diagnosis is very simple. Patient will recognize like color of urine is changing, then they will come to us. Okay. Treatment, it's not so dangerous. Generally, consumption of protein diet with low phenylalanine content is recommended. Okay, similarly, like what we have taken care in phenylketonuria, isn't it? Similarly, here also we have to take care. So similarly, we have other enzyme defects also, beta. Should just remember what is the enzyme which is defective and what are the clinical conditions.
Clinical. Yes, ma'am.
Albinism, no melanin pigments formation. Vitiligo. Leukoderma is, if it is seen in patches, you call that condition as leukoderma. Okay.
Yes, ma'am.
Then we have tryptophan metabolism. It's not actually very important, but if you feel like I will read everything or something like that. 99.9% it was never asked. Okay, you can
Okay,
also
ma'am.
cross check once. If you find
Yes.
anywhere like tryptophan, then you please learn this cycle, otherwise leave it. Chapter wise, you
Okay, ma'am.
write one test better. Because we have,
Yes, ma'am.
once we are done.
Yes.
Not, I don't recommend that. It's not required. Sulfur containing amino acids, methionine. Sam, I told you just now, right?
Yes, ma'am.
Methionine is essential amino acid. Cysteine is non-essential amino acid. So if we take methionine, automatically cysteine will be synthesized. And all these are the byproducts which are synthesized from it, which you can study like I have explained the previous amino acids. Now, for this answer, this is very, very important. These are, this side, whatever we have are methyl donors. Just now we have seen with one example in this chapter itself, we have seen norepinephrine. Sam has donated the methyl group, no? This was 2 carbon
Yes, ma'am.
compound and epinephrine was 3 carbon compound. Similarly, guanidoacetate is actually receiving methyl group from S-adenosyl methionine and then it is getting converted to creatine. Like that, you can just frame the sentences. This, all these things on left side you are finding these are methyl group acceptors, and on the right side of the table you are finding methylated product. Okay, acetinosyl methionine is giving the methyl group and the methylated product is formed. Okay,
Yes, ma'am.
and not only that, I want you to learn this also better, not formulas but just simply like methionine. It is now converted to S-adenosyl methionine in the presence of methionine adenosyl transferase. And SAM is converted to S-adenosyl homocysteine. Okay, methyl group is now moving from SAM to S-adenosyl methionine. So this is methylated product now. Okay. So further
Methylated.
conversion, you are finding adenosine homocysteine. Okay, from homocysteine again methionine. So this cycle is repeating. So methionine is receiving its carbon from one carbon atom, one carbon donors like tetrahydrofolate. Okay, and methyl tetrahydrofolate. We'll see how many more topics are there. One carbon metabolism. Only one carbon metabolism is important. Rest all are not so important. Okay.
Yes, ma'am.
No. So one carbon metabolism, what does it mean? So these are the amino acid like one carbon fragments what we see during metabolic reactions, which are able to donate one carbon, like methyl group can donate hydroxymethyl, methylene, methinyl, formyl. You have to memorize all these better, very important. Amino acid metabolism is particularly important for the transfer or exchange of one carbon units. The following one carbon fragments are encountered in biological reactions which constitute one carbon pool. So whenever one carbon is required, even SAM is requiring one carbon, isn't it? N5N10 formyl tetrahydrofolate is donating that carbon group, right? So everywhere where one carbon is required, any of these chemical substances will donate one carbon atom. That is the meaning. Okay. Shubhangi, are you there?
Yes, yes, ma'am.
So I want you to learn this cycle better, where you will get so many examples, in fact. Like see here,
Of one.
glycine and tryptophan, they are converted to formate, one carbon compound. So this one carbon compound will be able to transfer one carbon atom in the form of N10 formyl tetrahydrofolate, purines. Okay, they will be donating one carbon atom to purines, that is C2, and also formyl methionine. Similarly, you have like histidine is converted to formiminoglutamate, that is FIGLU. Again, this tetrahydrofolate is now converting this FIGLU to N5 formiminotetrahydrofolate. Whatever is given in the form of red boxes, these are active forms which are now donating carbon. And these are actually formed from these byproducts. For example, N5N10 is formed from serine in the presence of
Yes,
tetrahydrofolate and glycine. N5N10 is giving one carbon to glycine, serine, and thymidylate. Is that clear? But
okay. Yes, ma'am.
like that, you just go through. So that is one carbon metabolism. Now what you do is you just check in the question bank like what are the questions you have in this chapter, old, new,
Yes,
everything. Okay, and take one day and write question answers, because I hope you will have sufficient time, right?
yes, ma'am.
By tomorrow evening, you please submit. Now in the morning session, how you are studying? Better you tell me once.
Paper one topics I'm